So we're now into spinning the Krebs cycle, and as we said, the speed of the spinning of the Krebs cycle depends upon the two thyroid hormones. But the main function of the spinning of the Krebs cycle is to add 2 hydrogens to FAD, the active coenzyme of vitamin B2, and 1 hydrogen onto NAD, which makes it into NADH, plus an extra hydrogen there. So what we're going to do, or what happens here as we spin it, is we release the FADH2, which donates 2 electrons as a result into complex 1, and the NADH releases, with the extra H+, releases 2 electrons into complex 1. So what happens here, this is the inner mitochondrial membrane, that's the outer one, and remember that hydrogen is 1 proton and 1 electron. Okay? So the electron goes into complex 1, and the proton goes into the between the membranes. So complex 1 then hands on the hot potato, or the electron, to complex 2 via coenzyme Q10. And CoQ10 then hands it on to complex 3. So at this stage, we've got 4 electrons going on to complex 3. So we've got 2 from FADH2 and 2 from NADH plus H plus there. And as a result, we've also got 4 protons. Okay? So the concentration of the protons in the membrane is building up. And it gets to a certain point here when it then releases the electron and the proton together at complex 4. So it's like a pressure build, like an atomic nuclear generator here. So it's at complex 4 that these 4 electrons are added basically to oxygen. That's where the oxygen. So boom, all of a sudden oxygen has 4 electrons placed on it, which actually makes water. So our result of the machinery here is basically to produce water. But the energy that's given off adds the phosphate group to ADP to make ATP. So we get different levels of ATP being produced here. That's our high energy bond. Now, the point from a functional point of view and our interest is if these electrons don't go on in one go, but go on singly or univalently as it's called, we end up with reactive oxygen species. And this is how we make reactive oxygen species. Sometimes these are called free radicals, but not all reactive oxygen species are free radicals, but they're all reactive and stimulate inflammation. So if it's home in on complex 4 here, where it occurs, is we have oxygen, or molecular oxygen, O2. If we add 1 electron to oxygen, we get an extra electron on the outer orbit, and this is called superoxide. So this is the first of our reactive oxygen species. When we add the second electron, we get hydrogen peroxide, which doesn't have a dot there, so it doesn't have an extra electron, but is a highly reactive substance. So a reactive oxygen species, hydrogen peroxide, is a reactive oxygen species. When we add the third electron, we get hydroxyl radical, which is very reactive. And when we add the fourth electron, we get water and oxygen produced. Okay, so this is basically how we produce our reactive oxygen species. So when and why do we do this is very simple, that we wouldn't exist as a person to fight infections if we didn't produce reactive oxygen species. So reactive oxygen species are very important, but we don't want them produced too much if we haven't got an infection. So here we see another picture of the electron being converted from complex 1 to 2 to 3 and then into 4 here. Molecular oxygen being converted to H2O with the release of ADP at various and ATP. So that's basically how it works. So the whole of the point of the Krebs cycle is basically to produce hydrogens which can then split into protons and electrons, and like a nuclear factory ends up producing water from oxygen with a lot of high potency proton buildup in the membrane there, which activates the final bit of phosphorylating ADP to ATP. So these reactive oxygen species are a family or families of chemicals that use primarily by the immune system. So our white cells need to have something to spray over the infectious agent like the bacteria or the virus. So if we start here, normal mitochondrial oxidation. So this is what happens in the mitochondria, and maybe up to 2 to 5% of all oxidative reactions end up producing these free radicals. So instead of putting 4 electrons on in one go, it does it univalently, boom, 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 so it produces these reactive oxygen species in the process. So we do produce reactive oxygen species anyway, but to a small extent every time we produce energy. Now if we increase the production of energy by increasing our body temperature, if we got an infection, this is called a respiratory burst. So when we have a respiratory burst, we're basically increasing our body temperature, we're activating these enzymes to produce them, so we produce a lot more free radicals or reactive species in order to kill bugs because we're being invaded by infections. So when we got an infection, we get a higher amount. When we're detoxifying, so particular chemicals, xenobiotics, that have to go through phase 1 detoxification, which means hydroxylation with an OH group there, it produces 1 molecule of superoxide for every molecule of the toxin that's detoxified. So that's a lot of free radicals are produced, which is why people, when they live in a toxic environment, look much older because they've got a lot more reactive oxygen species or free radical reactions going on in their life. So if you're in a very polluted environment, you do tend to age much quicker. And finally, we produce free radicals or reactive oxygen species when we've got a lack of oxygen, as we'll see when we do the hypoxia module. So low levels or tensions of oxygen, and also high tensions if you were to be in a hyperbaric chamber. So people who go into hyperbaric to have what they call a dive, you know, if they've got multiple sclerosis and things and they can increase the oxygen by going in a dive, should always take antioxidants because they're going to increase the amount of free radicals produced, which is not a good idea if you've got multiple sclerosis. So divers and the people in high pressure chambers and things should always take free antioxidants. So hypoxia, which is very common, which is a low level of oxygen, stimulates the production of free radicals, as does infections and toxicity. So the first free radical produced was the superoxide, and this is then reduced, in other words, it has a hydrogen added to it, by a series of enzymes called superoxide dismutase, or SOD. So this reduces superoxide to hydrogen peroxide, and hydrogen peroxide will then be converted to water. And in order to do this, water and molecular oxygen, it needs an enzyme called, the favored route is catalase. So catalase is an enzyme here. Glutathione peroxidase, when we look at glutathione, we'll see is an important enzyme here. NAD and other peroxidasing enzymes can also reduce hydrogen peroxide to water. If these enzymes don't function, and especially in the presence of iron or copper, hydrogen peroxide will go on to make hydroxyl radical, which is one of the most damaging to the DNA structure. So we know DNA is like a ladder, coiled there, so you've got one side to the ladder and another side to the ladder, and if you get a free radical attack to the ladder, you get like a nick, it's called, in the side of the ladder. So if you get enough nicks on one side of the ladder will collapse on that side. But you've got the other side as a template to model the repair on it. But hydroxyl radical and peroxynitride here will attack both sides, nicks on both sides of the ladder. So if the ladder collapses, you've got nothing to model it on to repair. So these are very important, very powerful free radical reactions. Now the purpose of having hydrogen peroxide is that's sprayed by eosinophils, a particular white cell, to kill parasites. So this is the mechanism of how the white cells kill parasites, by spraying them with hydrogen peroxide. If we block this pathway here and here by catalase, glutathione peroxidase, or anything else that will block this, we shunt the hydrogen peroxide in the presence of chlorine or iodine or bromine into this pathway here to make hypochlorite, hyperbromite, or hypoiodine. And hypochlorite is basically household bleach. In America they call it Clorox, we would call it by trade names or whatever, but it's basically household bleach, which household bleach is always advertised to kill sort of 99% of all known germs. It doesn't actually kill 99%, but that's the mechanism of how certain bacteria are killed, mainly the gram-positive, but it does appear some gram-negatives may be killed by this pathway as well. So we need to switch this switch, hydrogen peroxide, to stop it going down to making water, to making hypochlorite, and stimulating this enzyme called myeloperoxidase. Now, this enzyme is stimulated by vitamin D, which is interesting here, and I'll find, and this hydrogen peroxide, this catalase, etc., is inhibited by colloidal or ionic silver. So this is how silver works as an antimicrobial. It has no effects as such into the microbe, but it just changes the switch here like a railway terminal, railway junction, to stopping hydrogen peroxide being reduced to water, and it shunts it in this direction. So silver, colloidal or ionic silver, very good for treating a lot of gram-positive infections, and particularly as a combination with vitamin D. So vitamin D and silver is a beautiful combination, particularly for treating gram-positives. Okay, now on this side we've got what are called reactive nitrogen species, and these reactive nitrogen species are made and derived from arginine. And we'll look later on, arginine is part of the urea cycle of excreting ammonia out of the body. So arginine here, in the presence of inducible nitric oxide synthase, INOS, goes on to produce nitric oxide, which is a gas, which is very good. It vasodilates our blood vessels, which is why they prescribe arginine when people need to vasodilate. But nitric oxide in the presence of superoxide, coming together non-enzymatically, go on to produce peroxynitrite, which is one of the most reactive of the species because it interchanges with the hydroxyl radical. So nitric oxide is the way we kill mostly gram-negative bacteria, which we'll talk about the difference between those later on. And nitric oxide will then go down onto peroxynitrite, which is for the real deep-seated intracellular infections like viruses and certain fungal infections. We find there's a need for more peroxynitrite. So the reason we have these is to boost our immune system. So these are the killing chemicals. So if we don't really attack the chemical, the infection properly, we move this over into the adaptive immune system and start building up antibodies, which we don't always want to do because they lead to the long-term autoimmune problems. So an autoimmune problem is an inactive, innate immune system, which is this. So we should be able to identify exactly which of the radicals the person needs and find the nutrients to open up the different channels. So we'll cover that when we look at infectious diseases in that module. So this particular chart here, you'll see now I've underlined certain enzymes. Those that are underlined are what are called heme-dependent enzymes. And heme is a substance made when we make hemoglobin. And we'll see later on in the hypoxia, very important pathway, because heme goes on to make a lot of other enzymes in addition to hemoglobin. A lot of detoxification enzymes, a lot of immune system ones. So we want to familiarize ourselves with some of the heme-dependent enzymes like catalase, myeloperoxidase. The phase 1 cytochrome P450 enzymes are all heme-dependent enzymes. So heme, as you know, supplies the iron molecule, as in hemoglobin. And that, therefore, the iron is therefore for an oxidative reaction. So it stimulates oxidative reactions. So here I just filled in, I think this is the one you got. So this one kills gram-positives, this one kills gram-negatives, this one kills parasites, and this one kills the deep-seated intracellular infections. So we talked about challenging for energy. So a weak muscle strengthens to magnesium ATP, a strong muscle weakens to magnesium ADP. For glycolysis, challenge for strengthening against pyruvate, which we've done. For the Krebs cycle, we challenge against NADH or FADH2. So that if we're going to challenge the Krebs cycle, we challenge the person with NADH and FADH2. And then you can challenge, if you want, if you're Virgo amongst us, you want to find out exactly in the Krebs cycle all the intermediates and exactly the pathway that's involved. But the answer simply is one of the vitamin Bs, usually, if we have a look at it in detail. And for the electron transport, you can challenge against CoQ10 is one way, but we can look again, complex 1 is NADH, complex 2, complex 3, and complex 4. So we have these different complexes in the pain diagnostic kits, so they can be challenged for, or you could use CoQ10 as a sample there. So now we've had a look at the energy pathway briefly. If a person strengthens to NADH or FADH2 here, we know there's a problem with the pathway of the Krebs cycle. So you could go back and say, well, let's test, starting in acetyl-CoA, let's test citrate, let's test isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxidoacetic acid, and so on. That's what the complete intermediates in the cycle are. And the same rule applies. Whichever one strengthens is the one that's needed. Go back one, and that will be the one that weakens. So that's the issue. But you don't really need to do that unless you really are enthusiastic about it, because they're all B3, B2, B1, alpha lipoic acid. Everything needs magnesium there. Some need a little bit of manganese. So we've looked at this one in more detail. We talked about lactic acid, tissues that function under hypoxic conditions. In other words, you've gone anaerobically, you've got no oxygen, if you like, functionally there, so you will shunt that into producing lactic acid. We find functionally the different sort of body types, which we call red, green and blue, tend to correspond with the types of lactic acid that are produced. So remember, there's the L, the D, and the DL lactic acid, and this seems to be related to the gene, to the profile, the metabolic profile of a person, and which hormone is the predominant one. So red people have predominantly thyroids dominating them, green the adrenals, and blue the gonads. Now, if you get a build-up of lactic acid in the muscle here, and we're producing pyruvate here from the muscle, we're burning glycogen, going to glucose 6-phosphate, but instead of then going into the Krebs cycle, we're moving it across to lactic acid. Lactic acid then goes from the muscle into the liver, and that will then convert back to pyruvate and back into glucose. And this cycle is called the lactic acid or Cori cycle, and principally needs a lot of vitamin B3 here. So if a person has a build-up of lactic acid, they need the vitamin Bs, but particularly the B3. So this would be something that cyclists, as I mentioned, because they're going anaerobic with their exercise, they're going to need a lot more vitamin Bs, because they're going to be relying upon the build-up of lactic acid as a source of fuel for them. So cyclists should always use a lot of Bs, and if you're taking the Bs, you might as well use a lot of magnesium, because the first pathway of making energy is all magnesium dependent. So again, magnesium, 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 and all the Bs, very, very important in people who are working anaerobically. So under anaerobic conditions, NADH cannot be reoxidized through the respiratory chain. Pyruvate is reduced by NADH to lactic, catalyzed by lactic dehydrogenase. There are 3 different forms, as we said, which form the D, the L, and the DL forms. The reoxidation of NADH via the lactic forms glycolysis to proceed in the absence of oxygen or anaerobically by regenerating sufficient NAD for another cycle of the reaction. Some tissues in the body derive much of their energy from glycolysis and produce lactic acid. So one of these is the erythrocytes, which is the red blood cells, the gastrointestinal tract, the retina of the eye, the brain, the renal medulla, and the skin. The liver, kidney, and heart usually take up lactic and oxidize it, but will produce it under hypoxic or lack of oxygen conditions. So some areas of the body actually can use lactate quite readily, and other ones, as you can see, can't. But this is again, is we need lots of vitamin B3 to help metabolize the lactate. So let's have a look now at the difference between aerobic and anaerobic. So aerobic, remember, means it needs oxygen. Anaerobic means it doesn't need oxygen. Anaerobic is principally the glycolytic fibers, in other words, the first formation of pyruvate there. And these are mainly white fiber in the muscle. So white fibers, when you eat chicken, most of the flesh is white, isn't it? Okay? This is because they work primarily anaerobically. When you're working aerobically, you've got oxygen, and therefore you need a lot of blood, because the blood is what carries the oxygen, so the fibers are red. So in a red meat animal, it's going to be aerobic. And of course, what we are is a blend of the two. So the person who works mainly anaerobically will have more white, and the person who works more aerobically will have more red. So in human muscles, it's a blend of the two. You get a certain amount of red and a certain amount of white, because we can work on both, can't we? So type 2 anaerobic ones are white. Usually multiple contractions for the first 5 seconds of exercising. So you're going to run off anaerobically down the track or across the road or whatever it might be as fast as your legs can carry you. So we use mainly creatinine phosphate there and then convert 5 to 10 seconds. Glucose is derived from muscle glycogen, is metabolized by anaerobic glycolysis, leading to lactic acid formation. We get a rapid depletion of muscle glycogen. So this is why you've got about 10 seconds that you can go anaerobically at top speed normally without producing the lactic acid. And if you can't metabolize the lactic acid, then that builds up, and then you get pain, and you just can't run any faster. So this is the Usain Bolt running 100 meters, okay, and then not being able to do it for much longer at that speed. Okay. Now, aerobic-wise, you use oxidative red fibers are predominant, and you get the first 4 minutes of blood glucose. So you got your blood glucose level up to optimal, and you've got about 4 minutes if you're exercising aerobically before you run out of fuel. At 4 minutes to 18 minutes, you start to mobilize glycogen, which remember needs the P5P, from the liver. So the liver is the first sort of source of glycogen. After 18 minutes to just over an hour, you start to deplete muscle glycogen. Okay, and then from an hour onwards, you're using adipose tissue, triglycerides, or fat. Okay, so this is the general sort of formula. So what is the difference here between these two? Anaerobic is the fast movement, so you use up your energy much quicker. In sport, we generally measure it using Phil Maffetone's figure of 180 with your pulse minus your age. So if you're 30, your maximum aerobic pulse should be 150. If you're 10, it should be 170. And if you're 60, it should be 120. This is your maximum aerobic pulse. So you exercise up to that point. So up to that point, you're working aerobically and you require oxygen. Okay? When you go above that point, you're then going into anaerobic. Okay? So then you would, if you can go on doing it, you need to go and burn lactic acid through the Cori cycle, or lactic acid cycle. Okay, so just work out how old you are. You might need your birth certificate if you're not sure of it. So minus it from 180, that should be your maximum aerobic pulse. Okay? So as you get older, it's nothing much more than a brisk walk, okay, or a gentle cycle ride. If you live in a village, there's always older people, like gardeners and things, who ride bicycles, aren't there? And they potter along at a very slow pace, but they seem to potter around for years and years and years. They don't do much work, but they're employed as retainers in gardens and things into their 80s. But they're working aerobically, okay? So they're exercising aerobically. And aerobic exercise is repeatedly now shown to help so many different illnesses, including last week a lot about cancer and things, and that it was very important that cancer patients exercise because it actually extends their lifetime, life expectancy. So, if you're a sports person or an exercise person, don't exercise anaerobically, certainly not too often, okay? So go to your maximum aerobic pulse. But if you've been ill or you're not used to exercising, don't go to your maximum straight away. Work on it for a few weeks, maybe 5-10 points lower than your maximum aerobic pulse. Remember that when you do go maximum aerobic pulse, you've got about 4 minutes. And at that point, because if you exercise at that 4 minutes, you think, I wish I hadn't bothered. Suddenly you sort of hit your first break, and then you have to go into your first reserve tank. Now, years ago, I had a Volkswagen Beetle car before the days of fuel gauges. Okay, so those cars, you just drove the car until it ran out of petrol. So you had to, every time you went to a fuel station, you had to write down what your mileage was. You knew roughly what the miles per hour was and how much it used, how much to go gallon, and hope that you filled your tank up. And I'm not the best of people, as far as filling the tank up. So I run it usually down to empty. But Volkswagen came up with the idea of putting a reserve tank in. So when you ran out of fuel, you turned this switch, which is always difficult to get to, and then started the car again, and you've had your reserve gallon of fuel there. And this is like liver glycogen is. So when you hit your first hurdle, it's as if suddenly you hit the brake on being able to run. So you run, you've got about 4 minutes, and all of a sudden you think, oh, what's happened? And you think, oh, I wish I hadn't bothered to come out. And then you get a second lease. Okay, then you go in and you think, actually, I'm doing very well. This is not too bad at all. And then you've got 18 minutes. Now, if you go on after the 18 minutes, you hit your second fuel, which is your muscle glycogen. So you're hitting your liver glycogen first, and then the muscle glycogen. So remember, to activate that, you've got to have adrenaline, or noradrenaline, so exercise is a stress, and pyridoxal 5-phosphate. So it's a good idea if you're sort of exercising up to 18-20 minutes or so, to top up your glycogen. Okay, so this is called glycogen loading or carb loading. So people who are going to do a lot of endurance exercise often load their carbs up the day before, so they build up their glycogen, but it's no good if you don't have enough pyridoxal 5-phosphate with it. So up to just over an hour there, you're then burning quite happily your glycogen. So at that point, you should then start to move into beta oxidation, which should be burning fat. Okay? What you don't want to do is to go back from here, if you're exercising for an hour, up to 4,000 minutes, which is a long time. If you're on a long endurance exercise, you don't want to go back to here. So the worst thing you can really do is to have a candy bar or a sugar drink. If you have a sugar drink, you'll flick back into here, and you've got to go through the whole process. So you'll come out of ketosis or burning fat and go back to burning sugar again. And that's the short-term fix. But the reason why a lot of athletes and long-term endurance sports people can't manage it. Once you go into ketosis and burning fat, you want to stay there if you're going to have to do a long-term like a marathon or a long swimming trip. So what you want to do is to make sure you get into burning fat. What you don't want to do, of course, is to start burning muscle. Okay? So if you remember, burning muscle or burning protein is mainly instigated by cortisol. So if you're in a highly stressed state, cortisol will be high, and you'll start to break down your proteins, particularly in your muscles like your hamstrings and things, much quicker. What you need to do if you're doing a longer exercise is to get into burning fat, and that aerobically takes an hour before it starts to occur. So this is all taken from Harper's Biochemistry. So we can test that. So anaerobically, we can do a muscle contraction at twice a second. So as if you're belting off down the track. In fact, it's better to do this, like a cross-crawl pattern, as if you're running down the track on the spot. And aerobically, you just do the same thing at once a second, so a slower mobilization. So if we go back there, we talked about aerobically making 38 molecules of ATP, 2 here, 2 here, and 6 there in the glycolysis pathway, making a total of 10. Another 2 in the Krebs cycle, so we don't make much energy in the Krebs cycle, but we make 28 molecules in the oxidative phosphorylation, that last pathway. But we've got to take 2 off for starting the program in the first place, was going to work, and that leaves 38 molecules in total from one molecule of glucose. So we produce a lot of energy from that. If you're working anaerobically, of course here what you're doing for energy is 2 from the first pathway, 2 from pyruvate, minus 2 to start leaves you 2 molecules of ATP. And then from here we got 1 minus 1 to activate glycolysis here. So we got 4, which leaves a total of 3, 4 minus 1, which leaves you a grand total of 5. So if you're working anaerobically, you only create 5 molecules of ATP. Okay, now you see why a person works anaerobically, they're tired. Okay, you cannot do more than 10 seconds or 100 meters of running now. So 38 molecules of aerobic and 5 molecules here with anaerobic. So a tremendous difference there. So we'll do a quick test now. Have we got anybody who'd like to do the test on the spot with the running? Shall we? Roger, did I see your hand go up? Right, Roger's going to come over here. See if I can test Roger's. Just to take a strong muscle here. If I take a deltoid, there, push up and over the head. That's nice and strong. Okay, you know, cross crawl like this. Okay, so run faster now. Run, 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 run. Okay, good. Give him about 10 seconds. Okay, stop. Okay, so that would be your contraction. Okay, and now run slowly. Is that good? Excellent. Okay, well, so aerobically he's fine, anaerobically he's fine, but let's put him anaerobic for more than 10 seconds, shall we? So fast one, but go, 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 get those arms up. Let's leave him there now, shall we? Do you think he's done the 100 meters yet? Well, okay, he's had it. Push. You can see he's had it. Okay,